Good morning, everyone. Today I'm going to talk about this article called A Novel 3D Polycaprolactone High-throughput System for Evaluation of Toxicity in Normoxia and Hypoxia. So the idea of this article was to prepare low-cost 3D culture matrix. And what I did was to use this polymer called polycaprolactone, which we will call PCL. And they prepare it inside a 96 well plate, as you can see here in the section A of this figure number 1. And in each of these wells, you can see here the fibers, and in this picture taken by the scanning electron microscope, you can see how the fibers allow the growth of the cells. So here you see one of these fibers, and the 3D matrix that is formed allows cells to grow in a setting that is similar to the tumor environment. So this setting is a 3D culture, and that allows to screen compounds in a better environment than a 2D culture. So here you see how cells have been stained in order to recognize GIMT1 cells with the CD44 to visualize in red the cells GIMT1, and in green, bimentin is indicated to recognize the fibroblasts. So 2 types of cells were cultured, and also 2 conditions were used. The first one is normoxia, and the other one is hypoxia. It's important to study hypoxia because in tumors, there are places where there is lower oxygen concentration, and that's why they use hypoxia in order to evaluate these 2 compounds. The compounds are paclitaxel and another drug that inhibits the growth of cancer stem cells. And they also culture these 2 cell lines. So here you see the picture of the co-culture. And the nuclei are stained in blue with the DAPI. So what they did was also to use the confocal in order to obtain images of the zeta axis, the zeta axis. And here you see 7 pictures and in the 2 conditions, in normoxia and hypoxia. And you see how the cells invade the PCL matrix, and in normoxia conditions, they enter and advance a distance of 98 micrometers, whereas in the hypoxia condition, they invade and enter 103 micrometers. Also, they present these dose response curves for the fibroblast, and also for here we see the fibroblasts, and under here below we see the GIMT1. So these are the curves, the dose response curves for each drug. So here you see in the 2D culture compared with the 3D culture, and here you see the colorimetric MTT, which correlates with the cell number. And so the cell number decreases according to the paclitaxel concentration. And here you see the effect with the 2D culture and the 3D culture. So we can analyze the compounds, how they behave in these different conditions and with each cell type. So this is an important contribution in order to study to screen the compounds, and it's especially made for labs with limited funding. Okay, thank you very much.